Thank you so much for joining us for our first or second summer speaker series session. I am very excited to welcome Dr. Steve Chang. He will be presenting on behavioral and neural mechanisms of social decision making. Steve Chang is a professor of psychology and of neuroscience at Yale University and a member of the Wu Tsai Institute and Kavli Institute for Neuroscience. He serves as the co-director of Yale's interdepartmental neuroscience PhD program and the co-director of the undergraduate major in neuroscience. His research program aims to elucidate the neural mechanisms underlying social behavior and to reveal insights into how these processes may go awry in disorders marked by social dysfunction. His laboratory employs high-density electrophysiological recordings across multiple brain regions during real-life social interactions to understand the neural codes, computations, and network-level dynamics that support naturalistic social behavior and cognition. You can drop your questions in the Zoom chat whenever they come up, and we'll tackle them during the Q&A at the very end. And I'll now hand it over to Dr. Chang.
Thank you so much, Cindy, for a kind introduction, and thank you for inviting me. I'm very happy to be part of this talk series. So like Cindy said, I'm going to focus on multiple types of work, kind of all point towards studying behavioral neural mechanism of social decision making at this talk. So in our lab, we've been thinking more and more about, studying social behavior with primate brain evolution and primate evolution in general in mind. And I think it's very informative given that, when you study social behavior, eventually what's guiding them is, in a way, social structure that different species evolved with. And even in primates, there's very diverse social structures. In our lab, we study two species, rhesus macaques, Macaca mulatta, old world monkeys that are competitive, but also studied quite a lot for their cognitive capacity in the field. But also in common marmosets that are New World species that are really great to model more like family style dynamics of humans because they live in family groups and they're very socially tolerant. So they're great for studying pro-social behavior and altruism, for example. So we're going to talk about both of these species today. So let's start with rhesus macaques. When you see these animals in the wild, they actually spend a lot of time grooming one another. Even though they're very despotic species, this kind of allo grooming is pretty important in their daily lives. And they also show cooperative behaviors. It's not as common compared to other species, but in the case of rhesus macaques, they're really good at cooperating to defend territories together. This is often called coalitionary battle. You can kind of see on the bottom right, one group is trying to invade the other territory, and they kind of work together to kind of, fight off the other troop. So they're kind of cooperative in this kind of antagonistic interactions. Marmosets, on the other hand, they're arboreal species living in Amazonian forests. They're much, much smaller, but they live in family units. So, and also, importantly, they're cooperative breeders. So it's not the mother who does all the rearing. A lot of father and also sibling members help out in the child rearing process. And they are pretty interesting in terms of studying pro-social behavior. As you can see from this plot here, if you look at the extent of allomaternal care, how much of the outside of mother you're caring for a child as a function of proactive prosociality, you can find this kind of representation in these two axes. Us humans are pretty good at both. Whereas macaques and chimps are very despotic species, and they're not that great at this dimension. Whereas marmosets are getting pretty close to the human level, so it's great to study, proactively pro-social behavior, and it kind of, fits into this kind of very interesting high social tolerance kind of group of animals. So before I go into the different sections of the talk, I want to first kind of highlight what is social brain, what we mean by social brain. Social brain is not the areas of the brain that are exclusively socially involved, but there are the regions that are commonly activated during all sorts of social behavioral tasks. So in humans, we see a lot in the medial prefrontal area, OFC, also reward-related area, hippocampus and amygdala as well. On the lateral side, everyone probably heard of temporal parietal junction. That's kind of heavily involved, and also the STS regions. When you go to the monkey brain, you see very similar structures in the medial prefrontal cortex, also hippocampus, amygdala, and OFC. But in the lateral side, monkeys don't have the anatomically homologous TPJ, but their functional TPJ seems to be more kind of included in the STS region. In rodents as well, you have kind of similar areas popping up in socially relevant brain regions, including ACC, DMPFC, amygdala, and hippocampus, and so on. So in the talk today, I'll be covering these areas that are kind of highlighted in different colors, the MPFC, ACC, both the sulcus part and the gyrus part, and OFC and the amygdala through some different sections of the research. So the outline is here. First, I want to first go over some findings speaking to separable but overlapping neural codes for representing decision variables for self and other when you're making social decisions, and then move on to how these different areas are coordinated in guiding pro-social decision making as opposed to anti-social decision making, and also how these coordination may differ when you're experiencing reward or when you're vicariously experiencing reward of another. From there, I'll talk about interactive social behavior in the domain of social gaze interaction, also in the prefrontal amygdala circuits. And the last two parts are going to be moving on to the marmosets, and I'll first talk about some of the new work showing that marmosets use really flexible and diverse social strategies to make working together work. And then add that, we found that one really good mechanism that marmosets use to gather social evidence to decide, when they should cooperate using based on social gaze kind of dependent strategy. So let's start with pro-social and antisocial decision making in macaques. So this task is kind of modeled after human dictator game kind of thing. And we imported that into monkey version of dictator game. And in essence, you have an actor monkey is looking at some symbols that they learn to map onto different reward outcomes. So it could be reward to the self, reward to the another, reward might go to both of them, or it might go to none of them. And there's a recipient animal who's sitting in the visible location next to them, just there to receive juice if the reward is going there. So in the past, we use empty chair as a control because you can have the exact same thing happening, except actor monkey might be delivering this reward to an empty juice bottle as a control. More recently, we kind of combined the two conditions so that actor can directly choose between giving to the juice to the partner or the bottle in a more efficient kind of way. But the really important thing about this task is that choices are always paired such that in the self-both context, for example, actor monkey can choose whatever they want and they'll always get the reward. But one choice will give juice to the other monkey too. Similarly, in the other bottle condition, whatever actor monkey chooses, he will never get the juice. But he has an option to be pro-social by choosing other. So the cool thing about this task is that because of that, we can measure their, social preference, whether pro or anti-preference, without varying your own rewards kind of perspective, which makes it interpretation more clear because you're not confounded by your own motivation. So sequence is goes like this. Monkeys usually fixate. There's some free choice depending on delivering juice to, let's say, self or both. Make a gaze fixation to indicate a choice, and then reward outcome happens. We also have this forced choice condition where the exact same kind of reward outcome is happening, but without monkeys expressing the choice. So in this case, monkey looks at, let's say, other symbol, and that's it. Experimenter decided this trial, other monkeys going to get the juice, for example. And there's a free viewing condition, so we can look at if there's some interesting gaze interaction happening at the end. We already talked about this. But when you do this task, we see a very consistent preference of the actor monkeys. So this is a preference index, as the contrast index of choice A, B over the sum. It's organized such that reward giving is higher, reward withholding from the partner is lower value. And here I'll show you both the social context and non-social control. And different colors here indicates the different partners, basically. So here, in the other neither condition or other border condition, and actors are systematically very prosocial. They rather give to the other monkey than wasting in a bucket, for example. Whereas you don't see that when you remove the recipient monkey from the room. And this variability of different pairs was explained by familiarity of the actor and partner, basically. Interestingly, in the self-both condition, you actually see the opposite pattern. They rather drink alone than drink together at the same time. I think there's some evidence pointing that that's because of they don't like drinking together at the same time. It's kind of competitive in some ways. But we see consistent bias towards the antisocial preference in that case. And this variability was kind of modulated by dominance. In an interesting way, actually, if you're more dominant, you're a little bit less antisocial in this case. And there's some work in the field supporting this, that dominant non-human primates, as long as there's resources not so restricted, they tend to share more. And again, you don't see anything in the non-social control. We saw this in many other studies in different data sets. You see the self-antisocial bias here, pro-social bias in other neither condition, and also in some other data set, we saw other bottle preference that they prefer other. In the self-both, monkeys tend to prefer self. So with that kind of social preference established, now we can look in the brain to see are there neurons that signal other monkeys' reward outcome? So that was the kind of goal of the study. So we studied the sulcus of the ACC and the gyrus of the ACC. This, for those who are interested in anatomy, this is 24A, B, and some of the 32. And also OFC areas. So in AC's gyrus, you see these types of single neurons that if you just look at the reward aligned data, you have increase in reward signal as long as as long as the actor monkey is getting the reward. This both reds are getting reward, choosing self over other or choosing self over neither, for example. But when no one's getting the reward in black or other monkeys getting the reward, this neuron didn't fire much. However, we also see a very unique neurons like this in AC gyrus that when you align to reward again, when self is getting the reward or no one's getting the reward, it didn't really modulate activity. But when other monkeys getting the reward by reward donation, you see this nice bump of activity. One other type of neurons we saw frequently in the ACC gyrus is that this kind of reward mirroring type neurons that as long as one of the monkey, actor in red or other partner in blue, gets reward, it's activated, firing rate increased, but when no one's getting it, it didn't increase much. So now we can now do the same thing in ACC sulcus and OFC and found some distributed patterns here. So for example, now I'm going to label proportion of significant neurons across these areas, and I'm going to call the neurons that increase activity or modulating activity only reference to self-reward as self-reference. But neurons that are like this one, increasing activity reference to other monkeys' reward, other reference, or this type kind of both reference. So one way to think about it is neurons like this is kind of self-regarding neuron. And neurons like this have some other regarding component there. So let's look at the proportion of these neurons. If you look at the self-reference neurons first, you have higher proportion in OFC, AC sulcus, but AC gyrus is much less compared to these two areas. On the other hand, now, when you look at the other regarding neurons, you kind of see the opposite pattern. So if you look at the other reference, it's highest here, both is also high. So if you group these together and look at it, self-reference neurons are decreasing, but other regarding neurons actually are a lot more in ACC gyrus. So it's separable, distributed, but there's some bias in area and how much you're signaling what happens to another animal. So are these neurons actually causally relevant? So we collaborated Betsy Murray at NIMH, and we lesioned the ventral part of the ACC. So a lot of these are include some of the sulcus, but most of them are in the gyrus actually. We modified the test a little bit to be able to analyze some pupil data, but the concept is the same. You have different symbols, and based on how much they're rotated, it tells you reward is going to self, other, or bottle, for example, and they have to respond it to get the reward. So what we found is that if you look at the control animals, and we're plotting the proportion of other trials completed in this case, so you can think about as prosocial preference other compared to neither here in the index. And three different colors are pre-operation, post-operation, and the post-operation retention of the learned one, and post-operation acquisition of the new symbols. So these are the controls. But what's interesting is that if you look at the ACC lesion animal, this is pre-op, so it's comparable to control. Postal retention is basically they learn this symbol, let's say this one, and then after surgery they did the task again. So it got reduced somewhat, but not that dramatically. But what's really interesting is that post-op acquisition, so after surgery, they learn the new symbol, new learning of new mapping of which reward goes to self, other, or bottle. In that case, with ACEs lesion, these animals just couldn't learn the new meaning in the other compared to neither kind of differently. They learned self totally fine, no difference there actually. But in that case, other and after the surgery, other and neither differentiation stopped with the new learning symbols. So it seems like, ACC is not retaining the memory of these representations per se, but it's really important to use the information from another to make the association that this is referenced to another animal. Human in the human brain as well, ACGRS has been shown to be involved in this kind of other reference cognition. So in the reinforcement learning, comparing high versus low reward probability decision for other engages this kind of other's reward signal correlates over in the gyrus of the ACC. But also, when you're listening to another individual's, let's say, advice to guide their probabilistic kind of choice, ACC gyrus in red seems to track this, how much are you weighing that advice of the other, whereas the sulcus portion, similar to the neural recording data, was all about your own history rather than listening to others' advice. So just the first summary of the first section, it seems that while social reward outcomes are represented in kind of distributed way in the prefrontal cortical areas, evidence seems to support that the gyrus, rostral, especially rostral ACS gyrus, seems to be a keynote in the primate brain for supporting vicarious reward and learning from what happens to other. So the next part with this, some of these basic representations kind of studied, we wanted to understand how these regions are talking to one another to guide when you're expressing preferences, but also evaluating the real world outcomes. So in this case, we focused on basilateral amygdala, BLA, and the ACC gyrus again. And we use this approach called spike field coherence. Basically, what we did is when animals are performing this same task, we recorded from multi-channel electrodes from both areas, BLA and AC gyrus, in the same hemisphere because they talk ipsilaterally. When you do that, now you get the spiking activity from one area as well as local field potential, LFP activity, and you also get LFP and spiking activity from the other area. And there's this coherence method called spike field coherence that can index how the two areas signals might be aligned. And you can look at how much the alignment or coherence changes as a function of different conditions. So to illustrate spike field coherence, imagine you are recording from both areas and you have this ACC local field potential LFP happening. And on top of that, you can kind of study when the BLA neurons are firing at different phases of the ACC's LFP. Similarly, you can also record LFP of the BLA and trying to understand, trying to discover when does ACC gyrus neuron spiking at which frequency and which phases of this BLA field. If there's alignment, then you will have high spike field coherence that might index there's some coordination going on. And to us, this is a little bit better than field coherence, just correlating two LFP signals, because those are a little bit more susceptible to the third common input problem. If there's another area sends both, then it can cohere very easily. Spike field is a little bit harder to be driven by common input compared to field because the time scale is just so different. So because now it has a frequency domain, we're plotting, spectrogram here across frequency on the y and time from decision on the x-axis. So time zero will be basically time of monkey indicating a choice in this case. Because we can have two different ways to align spike field coherence. On the left, I'll be showing you BLA spike to a BLA spike and ACC field coherence, whereas on the right side, ACC spike and BLA field coherence. Let's look at the first one first. And you can see here around the time of making a choice, you see that there's an increase in spike field coherence around the kind of beta frequency range within the time of making a choice. And this is a difference in coherence, so that we're comparing when monkeys are making prosocial choice versus antisocial choice. And again, because the reward is matched, we're not subtracting whether you are getting reward sometimes or not getting reward. Um, so that means there's an increase in coherence in this beta channel when monkeys are expressing prosocial compared to antisocial preference. Interestingly, when you look at the different pairing, you now see a shift away from this beta range to more like a gamma range. But also timing is a little bit delayed compared to this one, right? But nonetheless, this again shows the increase in gamma coherence between BLA field and AC spike that's greater for prosocial preference. Um, I mentioned about there's a forced choice in the past. It's very interesting to me that all these differences are exclusively found when monkeys are actually expressing the choice. So when you align to the same cue that predicts the same outcome, you don't see significant difference between prosocial and antisocial preference. So this means that somehow this coordination that are frequency specific and pairing specific, right, seems to be really unique to when monkeys are expressing choice, voluntarily making a choice compared to just knowing the knowledge of this reward or that reward is happening. So when you look at this kind of coordination in this task, you can also think about reward outcome because sometimes, if you choose to reward yourself, there's self-reward, which is studied a lot in the field. But when the reward goes to another animal, that has not been studied as much. However, these animals are keep showing to show social preference in certain contexts. So there has to be some reinforcing aspect of that. So for that reason, a lot of people call this kind of vicarious reward. So here we also have a chance to compare this kind of coordination pattern between experiencing and vicariously experiencing reward outcomes. So now it's the same way of showing the data, but now I'm plotting the time from reward onset at the x-axis. So time zero is the reward onset. Same thing in the frequency. And now the contrast has to be a little bit different because you're comparing reward outcomes. So this is self reward minus bottle reward and other reward minus bottle reward. So when you look at this pattern in these two contexts, you see that the kind of alpha beta-ish frequency coherence has increased, receiving reward for self compared to other. But when you look at the more gamma range, it's actually opposite. There's a higher coherence in the other vicarious reward condition than the self-reward condition. So this is shown here between these two. And you can kind of unfold that into two dimensions and see that at the time of reward, for that frequency, coherence was higher for vicarious reward than self-reward. And this was reliable enough that you can actually decode who got the reward from a simple linear decoder compared to null. Now, when you go to this lower frequency that I mentioned before, the pattern is opposite. So now you have a higher coherence for self-reward, but no change in coherence for vicarious reward. This is also reliable because you can decode it over the null. What's interesting here is that we're not looking at different pairing, like expression period that I showed you before this one, but this is fixing the ACC spike and BLA field in the same neural population, same neurons. But interestingly, even the exact same neural populations are showing different channels of coordination as a function of experiencing reward or vicarious reward. So that was interesting to us because you can kind of think of it as same neural pathway in some ways, right? But the which channel information might be communicating could differentiate two types of reward outcomes. Um, the spike field coherence actually doesn't tell you about directionality. So it just tells you are they like coupled together, right? So we have to also look at, you have to do a different analysis to look at directionality. So we did partial directed coherence. It's based on Granger causality method. When monkeys are making pro-social preference or anti-social preference in the preference time, making a decision time. And as you can see from this plot already, if you look at the difference in PDC between pro-social and anti-social preference, you can kind of see that, when you look at the information going from BLA to ACC, you have this strong directional component in the beta frequency coming up earlier, and then time shifted a little bit, right? Is similar to gamma that we showed earlier. And the gamma coherence area, similar to that, you have a high directionality there again. In the opposite directionality, there's really not much specific thing going on. Um, on the other hand, if you look at the antisocial preference, it just doesn't have this kind of specificity that you see in the prosocial preference in these circuits. So now you can kind of plot this over time, difference in PCD over frequency ranges. And if you look at the BLA to ACC direction, which is these guys, right? You can see that there's an increase in directionality at the beta frequency range and then another one at the gamma frequency range compared to information going in the other direction. On the other hand, when you look at the reward evaluation period, we found kind of different patterns. Here, if you look at the partial directed coherence again, and we're going to look at the gamma period first. This is where we saw vicarious reward related coordination increase. And what's interesting here is that regardless of self, other, or both, directionality is stronger from ACC to BLA compared to BLA to ACC in all these reward outcomes. Experience reward in the exact same analysis. Now we're looking at kind of alpha beta frequency range, and you see similar thing. ACC BLA is dominant, more directional flow compared to ACC to BLA. So it seems like information flow directionality and frequency channels used in this kind of coordination seems to be different depending on what phase of decision making you're in. Are you making decision or are you evaluating the outcome? And it seems like in this task, monkeys are not learning new thing every time, right? They just know what they prefer, so they're expressing it. In that case, BLA seems to be driving more medial prefrontal areas like AC's gyrus, whereas outcome phase, you're kind of evaluating and maybe adjusting your next choice and things like that are more part of like a learning mechanism in some way. In that case, it's consistent with the observation learning literature in rodents, for example. ACC seems to be really driving the BLA neurons in there. So to summarize this part, it seems like the neural population in the social brain areas coordinate activity to guide these pro-social or antisocial choices. And there might be this kind of frequency module. So if you think about orchestra, you have multiple sections, you have to coordinate somehow, right? And depending on, what the goal is, it could be pro-social or antisocial, how you're coordinating at different frequency module might be relevant there. It's just an analogy, but seems to be like a pretty intuitive analogy in sometimes. So far we talked about explicitly making decisions, but social gaze is also another important component that kind of links social decision and the social attention kind of together. So here's that video again when the two troops are fighting. So there's one troop and there's another troop. You can kind of see they're looking back to see who has their back. I think that's really important. So they're checking, do you have my back, my friend? And then he has his back, so they kind of, work together to fend off the other. So social gaze is very important in their daily lives. But we don't know much about social gaze processing in the prefrontal cortex. That's more in the action side rather than just a perception side. So we decided to study social gaze interaction in a more naturalistic context uh, that are not task constrained. So in our case, two animals are just sitting face to face, and they can spontaneously interact. Whatever they want to do, they can do. And we have some non-social objects like toys or banana hanging in the corner as a comparison point. And we tracked eye position in both animals at 1,000 hertz. And M1 in this case is the recorded monkey. And importantly, to calibrate, these animals really well, in addition to the typical eye calibration you might do on a screen, we also did this template calibration. So basically, behind this LED panel, there's another monkey there. So you can actually map out in that depth how closely all these points are there. So you can now exactly know, are they looking at the eyes or the mouth or right next to the eyes and so on. Just to show you the spontaneous gaze preference, if you look at the duration of each average duration of each gaze, monkeys look at eyes longer, followed by non-eye region of the face, followed by object. And also, if you look at the number of fixations, same pattern, they look at the eyes the most, non-eye part of the face and object. And this is kind of expected in this task. That's the most interesting dynamic region in a way. Like eyes, actually, monkeys look at eyes a lot. They oftentimes don't like making mutual eye contact for too long because that can induce aggression. But their interest is eyes, still predominantly eyes. So they tend to look at the eyes when other animals slightly not looking and so on. So in this kind of paradigm, we recorded from multiple brain regions in this prefrontal amygdala areas, AC gyrus, the MPFC, OFC, and the BLA of the amygdala. And these are just some recording areas to see first what are these neurons doing in this kind of spontaneous social gaze interaction. So I'll first show you some example neurons color-coded by what they're looking at. And it's interesting to keep in mind, there's no task structure. Other monkeys are not like appearing out of behind hidden scene. They're always there. So if a neuron is representing looking at the eyes, for example, it can start representing long before they look at the eyes because other stimuli is always up there. But here's an example of one neuron in BLA. We call it categorical social neuron. So if you look at these traces, time zero is the aligned to the looking at that stimulus, right? And you see that as long as monkeys are looking at the, face, non-eye face, or eyes, those are three colors, they increase activity. But when monkeys look at the object, it kind of decrease activity. So it kind of seems to be categorically grouping social and non-social. We also saw neurons like this. We just call it graded. Monkeys preferred eyes the most. Well, this neuron has the highest activity for the eyes, followed by face, followed by non-eyes of the face, and much less for the object, similar to their behavior preference. Similar to maybe this neuron might be tracking some value in that line of thinking. And there's also neurons that are really object specific. These neurons really have a very strong, sharp response just for objects. Um, we saw these types of neurons not only in amygdala, but also in the other areas. So OFC, you have these categorical neurons, graded neurons, object selective neurons, ACC2, categorical, graded, and the object specific, as well as the MPFC. So it seems like these social and non-social kind of representation seems to be very widespread in these brain regions. One thing that I wanted to highlight from this work is we looked at social gaze monitoring because they're spontaneously looking around. We can kind of ask, are they actually caring about where you're looking at in a moment by moment basis of the another monkey? But also, do these neurons track moment by moment what other monkeys looking at, your eye region? So we calculated this something called self distance. Basically, if you look at this from the M1's perspective, and this is M2, the other animal, you're just fixating, just all around the place. But every fixation, you can calculate a Euclidean distance from that fixation to other monkey's eyes, right? And if you're looking at the dead on at the eyes, there will be zero, for example. If you're like far, there will be big value. So you can calculate for every fixation all the Euclidean distance to see, do neurons care about this? Um, so what's shown here is aligned to that individual of those each fixation and separated by if that distance is small to large in four different levels. And here's an example neuron in OFC. This neuron increased activity and scaled activity as a function of distance, such that when it's closer, it fired more, but when it's farther, it fired less. And you kind of look at the average and you can kind of see it's kind of monotonic nicely. You also saw neurons that are opposite, that fires more when you're farther away than when you're closer. So you see the opposite pattern of monotonic activity here. You see those in DMPFC, ACC, as well as the BLA. So these are pretty interesting fact that you're continuously tracking how far are you to the eyes. You can think about this as like a dynamic attention level or monitoring my own social gaze and so on. But what's really interesting about this data is that you can also look at what other monkeys are looking at you, how they're looking at you. So you are now the M1 recorded monkey, so there's being recorded. You know how far, if someone is paying attention to you by looking at the eyes or looking away, but you also know how far are they looking away. So you can do the same thing where now the Euclidean distance is where other monkeys fixating relative to my own eye region. So it can be close or it can be far. And we see neurons very similar in those cases too. These are like different neurons than these neurons, but you see that this neuron actually had the highest activity for when other monkeys looking closer to your eyes or looking at the eyes, but decreased monotonically as they look farther. But this neuron showed the opposite pattern. And you see that in neurons that show this kind of pattern in different areas as well. So this kind of social gaze monitoring seems to be also pretty widespread in these areas. So I didn't have time to show you everything, but we actually looked at three different types of signatures here: social discriminability, face versus object, and also are you discriminating eyes versus mouth. This one we just talked about self-distance and other distance coding. We also looked at mutual eye contact selectivity, where does the looking at the eyes differ when you're making mutual eye contact or looking at alone. But the reason I showed you this slide is that there are some neurons that are doing one of these only, but there are other neurons that are doing more than one of these. So this is like the same idea of a mixed selectivity that's kind of gaining traction in the field. You said that many neurons are overlapping. You can also further quantify, are there differences in these brain areas of how the mixed selectivity is instantiated. So, for example, in OFC, neurons that encode one or two, neurons encoding one or more than one variable are kind of similar in proportion. Same thing in the MPFC. But in ACC gyrus, neurons encoding one was actually greater than in proportion than neurons encoding more than one variable. Whereas the BLA is actually the opposite. Neurons encoding more was less than the neurons encoding more than one variable. So there's a kind of varying degree of mixed selectivity in these regions, even though representationally there are a bunch of neurons doing these similar things out there. So some of the mixed selectivity use seem to be somewhat different there. And the final part of this section, I want to show you that can we actually use this knowledge to increase or modulate social attention. So in this experiment, what we did was in the exact same setup, we kind of detected in real time whenever monkey look at other monkey's eyes, we delivered closed loop real-time microstimulation to either orbitofrontal cortex, the MPFC, or ACC gyrus to see if that momentary feedback stimulation will change how they're going to look around in the next 5 to 10 second period before they go into the next sham or real stimulation. We also had a non-social control condition where we just put a iPad kind of screen in front of the other monkey and show two random dot motion stimuli because they're salient to them, so they look at it. And we can do the same thing. Whenever they look at the random dot motion stimuli, they get closed loop microstimulation in one of those three brain regions. It's worthwhile noting that we use low microstimulation because field has shown that if you actually use higher one, you can more likely to disrupt the signal by injecting noise. Lower one, especially in OFC, has been shown to kind of facilitate some of the computations in decision making, for example. So we ended up using a very low 75 microampere in 100 hertz, just a short time, and soon as the monkey look at the eyes. And also, it was 50% real stimulation, 50% sham stimulation. And importantly, stim and sham were at least 5 seconds apart so that there's a time to actually look at what happens after that stimulation. So this shows the kind of summary of the spatial aspects of attention. So what this is showing you is that imagine there's another monkey here, and this is the eye region, this is the face region, and this heat map shows the effects of looking at those area steam compared to sham. So basically, whenever you see your kind of red increased heat there, that means they looked at the eyes more following steam compared to sham. And this is not the stimulation, not the fixation that triggered the stimulation. We actually didn't see any effects of that. They didn't make the eye stick or they didn't repel the eyes or anything like that. The saccadic kinematics were also totally unchanged due to this stimulation. So this is showing you the uh, subsequent fixation in the next 5 second period. And you see that after OFC closed microstimulation, they look much closer to the eye region. It's not that they always like move eyes direct, always look at the eyes. They just distance is much shorter, so they concentrate more towards the eyes. Whereas we didn't see that for the MPFC and ACC. And you see that really nicely for each animal. If this is one of the monkeys, monkey L. If this is another monkey, monkey T. You also see you don't see in other two areas. There's also effect on the temporal dimension, which is also relevant for social attention. So we looked at a couple of measures, such as interlooking interval. What is the interval that you look at other monkeys' face or eye regions? If you're attentive, that will be shorter, for example. After microstimulation to OFC, but not the other areas, we see that interlooking interval significantly decreased in both animals. Another thing is reciprocation latency. So what this is like if I'm the stimulated monkey and let's say another animal look at me, then I wasn't looking at the other monkey, right? But how long it takes me to look back at the other monkey. So if you're being watched, how long it takes for you to respond, which is called the reciprocation latency. And we compared how does that latency change after the stimulation happen. Here too, just in OFC, but not in the MPS and ACS gyrus, we saw reduction in reciprocation latency after OFC stimulation. So this suggests that both spatial and temporal dimension, this kind of super light microstimulation that doesn't change saccadic behavior or kinematics at all, can actually increase your looking behavior to that behavior that are tied to the stimulation. So it's almost like as if assigning a little bit more value to that looking target, for example. Importantly, we didn't see any of these in the non-social control. To just to show you that it's not that just anything you look has you can change it, but it has to have some value, maybe intrinsic value to it. None of these effects were different in the random motion stimulus condition. So the final thing is in the social gaze domain is, we can also think about breaking down social gaze into different primitives. So, whenever each gaze you make, you can have a content, what state you are in, you can be highly engaged, low engaged, or duration. So you can actually break this down in each trial. So this is an example trial, example session. Some sessions you look at the face a lot more than object, but other sections it might be opposite. So you can actually calculate some social state, engagement state based on these larger time scale windows. And when you look at this, you see there's some variability across sessions, session 1 through 41, let's say. And then you can kind of do some clustering to identify, which were high state, which are low state. This was kind of median split way of doing things. And then what we found that is these components, are you looking at face or object or high or low state? Um, if you model that to see if those state of the M1 can change M2's behavior, you can. So, for example, here, if you look at the M1's content and duration or state and content, for example, those log likelihood odds is less than zero means that there's an interactive effects on M2's looking back behavior. So there's some function of how am I combining these interactive effects of these combined primitives can have a different looking behavior effect. Um, Neurally, we wanted to test are these related to some kind of compositional computation in the brain? So we can look at, this kind of test. We can generate this kind of compositionally hypothesis where you have content and state. You can have high state, low state. Are you looking at face or object? And you can examine, can you generalize states across different content, or can you generalize states across different content? So we tried this, and we found that these neurons in these areas, BLA, CCTM, PIPS, OFC, they all have separability of state and content. So it actually is one part of the compositional hypothesis we wanted to test. They all passed. When you go to the orthogonality at the geometric analysis, we found that BLA and ACC gyrus it was very orthogonally representing state and content, whereas DMPFC and OFC was not as orthogonal like BLA and ACC gyrus. And finally, the generalization that I told you about, BLA and ACC gyrus showed both content generalization and state generalization, but DMPFC didn't show content generalization. OFC didn't show state generalization. So it seems like some of this area, especially BLA and ACC, kind of meets all the three criteria that we test for compositionality. I'm going to skip this because of time, just to show that there's also directionality of interaction depending on content and state. I'm going to skip this due to time, but I'm going to move on to the final section of the talk. They're looking at now marmoset behavior. So we wanted to study cooperation in marmosets because they are very prosocial animals. And traditionally, cooperation have been studied in this kind of rope pulling task. So chimps are pulling ropes. These trays are very heavy and chimps are very strong, so they're really working hard to pull the lever together. This has been shown in many different species, and animal behavioral field has really liked this paradigm for many reasons. But interestingly, in this task, you can see that when one animal is less motivated, this animal is soliciting behavior. So you can see that soon again. It's heavy. He's gonna turn around and he's like, "Come on, let's do it," right? So that really exemplifies. I want to work together to make this work in this task. So that's kind of types of cooperation behavior we wanted to study in marmosets. So we developed this marmoset apparatus for automated pulling called Marmo app because when you do rope pulling kind of task in marmosets or other animals, they maybe do 10 trials a day per whenever they come out to do the test. So they don't do enough for neuroscientific experiments. So here in the plot, you see that this red line is like mean number of trials across all the papers out there we can find. It's about 10, 15 trials per session. Whereas when you have this kind of automated pulling paradigm where they have to pull, marmosets have to pull the lever to cooperate, and you can actually adjust the force level to make it easier for them and so on. You can go to about 150 trials per session for self-reward and also cooperation, whether successful or not. So now we can have enough N to start analyzing behavior and neural data. Just to show a video of what they do in this task, it's pretty intuitive. This is the levers. These are two animals. Reward is coming out here. And when you watch this, look at kind of their gaze behaviors. Success, drinking, and then they're pulling, look at each other, drinking. So we do this training starting from self-pulling first, and then pull within 3 seconds to be successful. If they're 50% success out of all the pulls, 50% of the pulls are successful, go to L2, 2 second condition, all the way down to 1 second condition. And that was just enough to show that they're they have time to look if they want to and pull it's just a nice time, functional time window to study this. And we had two kind of controls. No vision control. What happens when you block the total view of the other's levers too, to see how much vision is used. And also auto lever control. So we just block panel here, and this animal is gonna pull with automatically moving lever according to this animal's prior schedule. So we wanted to see, are they just using variable schedule pulling, or are they actually can time with this non-social object? So here, these are the two conditions. And what we did was mutual cooperation, no vision, mutual cooperation again, automated lever for more than like 10 days, mutual cooperation again, just to see how the behaviors change. So here's a no vision control. You can kind of see they feel very out of context, right? And these are well-trained animals, but they're just having really hard time. Something's really off because they're using gaze. It's just part of their behavior repertoire in this case. Sometimes they get successful. Here's the automated lever control. It's blocked here, and this is moving automatically. But these animals are pulling a lot, and it's not necessarily, well, this time it worked, but this will move along too, right? So there's a lot of extra pulls that didn't come to rewarded. So you might actually have a lower success rate. So when you look at the data, success rate over time of this thing I showed you, you can see cooperation, 50% is what we targeted for, and then it drops with no vision. It doesn't recover that much, and then go back up for mutual cooperation. And once you go to automated lever, it drops and stays dropped for like 10, 12 days. So it's really interesting. They don't come back. They just have a hard time reacting to the automated lever as a way to cooperate. This becomes important for the neural study because we're finding that the complex behavior of prepared behavior of other animals' action is serving all the cues that are necessary for the animal to time it and pull. So they're just having a hard time matching the final output of the non-social object. So success rate clearly shows these differences. Um, Interpool interval is also different. So interpool interval is much shorter for mutual cooperation compared to other two conditions. And this is including all the pools, not only successful ones, to see how they're coordinating tightly. And reward count is also higher here, but much lower for no vision and automated lever control. So with this, we can also use, this kind of deep lab cut tracking method, markerless tracking method combined with AnyPose to monitor what they're looking at during this behavior. So he's looking at one another just now. And once we do that, we can actually do an orthogonal projection to see what they're looking at during this behavior. And marmosets actually use a head gaze as a main way to socially orient. Social gaze is their head gaze. So it makes our life much easier here. So now we can see what kind of social gaze strategies are used, what's social gaze for. So when you look at the social gaze rate here on the Y and success rate, it's actually negative correlated in kind of big time window. So for example, when monkeys are not doing well, they actually use social gaze more. You can kind of see that here. Successful pools compared to failed pools, general social gaze rate increases. It's almost like using social gaze to coordinate when success rate is high, they reduce the reliance on that. So that's one finding. Another finding is that when you actually time lock to other partners pooling, you have a different time resolution of looking at social attention. So in this case, if you time to partners level pool on zero and do cross correlation, you see that successful pool is much more time locked than unsuccessful pools, which means that this is like a temporal precision issue that if you're very good at monitoring when others are pulling, that gives you confidence that if I pull, it will be a successful cooperation. So it seems like there are two time scale of social gaze thing going on here. One is kind of vigilance mode. Are you using it to make sure if you're doing well, you don't have to use much, but if you're not doing well, you might use to coordinate. And this kind of precision coupling mode where you are coupling exactly when other monkeys behaving to know that it was successful. Now we can use a dynamic Bayesian model to model all this because now we have time series of animal ones pulling, time series of animal twos pulling, time series animal one social gaze and animal two social gaze. So this is unsupervised graph method to discover causal temporal structures of these four different nodes here. And then it goes through and discover, and it gives you some kind of edges. And what's cool is that that edge can actually tell us what strategy animals are using based on this. So for example, what we found is in this strategy, we found pull-in rhythm strategy. This is expected because tests required it. But for example, animal one pulls, animal two pulls without looking. So dependency error might be this way. And this is social attention strategy. What animal one's pulling, animal two's looking at that. So you have this kind of directed graph. In this case, it's more like gaze lead pull strategy. I look at you and then I pull. So you have this kind of dependency structure. So we discovered this and we tested, are these actually functionally relevant other than being reliable to be discovered? And you look at when we look at the modulation index in mutual cooperation for successful or the failed pools, and you can see that both social attention and gaze lead pull strategy is used more when you're successful. You can also test generative model version of this to see, can you predict pulling actions? So launch strategy at the end can predict about this much, about 75 or 70%. If you just use pull-in rhythm strategy, prediction goes down, but still above chance. If you look at just gaze and pull strategy, prediction goes down, but still above chance. But now, when you just put together these two strategies that we extracted, it fully recovers the fully learned strategy. So even though they might use multiple strategies, at least these two are robust enough that when you combine it, you can kind of recapitulate the launch strategy. So it can also help predict the behaviors. Finally, if we're really thinking about studying cooperation, it's important to know, is this really adaptive behavior? So what we did was we changed a partner A, B, A, B, A, B across 10 days to see how they're adjusting. The reason why we did that was partner A was really not as good. Partner B was really good. And you can see the target animal's behaviors completely change. And I'll show you here. So now Ginger is playing with Dodson. Ginger is very good animal. Dodson's not so good. You look at Ginger's always pulling twice for Dodson. Uh, why twice? Pulling twice actually cues the other animal, but also increase the cooperation window to more than 1 second. So if the other animal is not as motivated or slow, you can actually make that more successful. But now, same animal is going to play this task with another animal named Kanga, who's actually very good. Now, you never see the double pull anymore. The ginger pulling always once, right? Sometimes ginger even follows Kanga. In this case, follow Kanga, and ginger's just pulling once, right? And they look at each other a lot more here too. So, and this was repeating back and forth, right? So we can see the pulling ratio go up and down across these days. You can quantify that. And also, when you're working with monkey that are not good, Dawson, you actually looked at the other monkey more to kind of make sure to pull twice at the right time for them to signal and so on. So this is flexible strategies that are partner dependent, which was important to know because they're trying to make this work. So what neural mechanism guide these gaze dependent strategies? So we had a hypothesis that maybe social gaze is a way to gather some evidence from another partner to figure out this is my time to make a decision to cooperate. So we designed this a wireless recording system. You can see in both animals here, they're working together while we're getting signals. We recorded from DMPFC, Brodmann area 8B and 9. Um, so going back to this tracking, because we're interested in social gaze as some kind of functional behavioral tool here, we first defined functional time window. And this was defined by what are the interval ranges? It's usually about 4 to 10 seconds. And this kind of definition becomes very important here because there is no trial structure here. So we need to think about how to analyze the data in this kind of way. When you look at the lever pulling behavior and social gaze looking patterns, we saw something interesting. So when you turn social gaze into probabilistic curve, you can see that social gaze density peaks right before time of pulling. And this was true across all many social gaze and pooling behavior. This is the pooling time for each animal's perspective. Social gaze was peaked right before pooling time. So we thought, well, there's something functionally meaningful here, maybe that social gaze being peaked may be related to decision to pull. So how do we decide what is trial start? If you want to think about accumulating evidence or something, you need to know when is it starting. And there is no trial structures. We have to think about how to define it. So we looked at all these different variables. I'm not going to go into naming all this, but you see that many variables are very all correlated, right? While they're doing this behavior. So which one is still the best predictor? Um, so we used GLM kernel method to discover which one has the most variance importance using drop in ROC area under the curve analysis. Also, which one has the variance inflation factor value. So it turns out in both animals, this LS, linear speed of the actor, was pretty good in terms of variance importance, but also it has the lowest variance inflation factor. So we kind of decided to go with that, and you can kind of see why here. So this is just showing linear speed, and you can see the linear speed kind of increases in the beginning and peaks, and then there's a pull and goes down again, and it will come again and peaks around the pull. Another way to show is this is motion energy plot. You can kind of see that face area motion starts early, and then later is the pulling energy, right? So if you are monitoring another, you actually want to use the face to start knowing the other animals approaching there. And this is drinking, and then they reset and start approaching. That's the linear speed of the face. Um, when you do that, we actually see a pretty nice correspondence between pooling and linear speed change. So you can think about when the linear speed is low, that's a trial start. Right? And then linear speed goes up and there's pooling and so on. So now you can define trial start to the pooling as a response time. And you see similar distribution across that, and which is very important for modeling, let's say, drift diffusion model. So the second thing is that, we wanted to model this drift diffusion model of are you getting to some accumulation point and deciding to cooperate, but what are you accumulating? So in this case, we took a holistic approach of partner's behavior. So because we did a PC1 of all those 8 variables that I showed you, and PC1 does a pretty good job with it. So we kind of call PC1 as action partner. And now, can we model this drift rate based on my linear speed and action partner? So I'm showing you the winning model. We tried many different models, but this is the winning model. Drift diffusion model is modeled by linear speed of the actor and the noise term, and partners PC1 of the partner, right? And the noise term of the PC1 of the partner. And importantly, there's like a given gaze actor here. So this was the only best fitting model as long as this term was there. So including PC1 partner all the time is not going to make the model the best. PC1 partner was only used in a productive way in the modeling side whenever actor actually look at the other monkey. So it's really like active gazing, active sampling of information is important rather than information just coming in all the time. Right, that's that. And when you look at this term here, the noise term of PC1 partner, that was the most interesting one. So basically, what it shows is that when the noise term is low, social evidence was greater, so drift rate was higher. When the noise term was high, the drift rate got slower. So social evidence is basically 1 over that. So we can now think about this PC1 or partner's noise term as a social evidence, like an uncertainty kind of way. So here are the actually neural data from the MPFC. You can align the data to pool, for example, and I'm breaking down by slower to fast reaction times. And you can kind of see the fast response time as high slope, then medium, and then high, as you expected from the drift diffusion model. And in fact, all the neurons have this negative correlation coefficient there, meaning that, evidence accumulation dependent on response time in the right way. You can also align the data to trial start based on the linear phase speed. And now you can kind of break the data down by high, medium, low social evidence. Again, this is 1 over that noise term of the partner PC1. And you can see that high evidence is higher slope compared to medium or low evidence. And you can see all across all the neurons generally now that's positively correlated. Just as a population summary, I just want to point out one thing that all the neurons that encode this social evidence slope is part of the neurons that encode this response time slope. So there's overlapping computation, which is a good thing to argue that these neurons are doing some kind of evidence process. Um, and you can also model this in neurons directly. So you can model neural DDM to slope, neural starting point Z just firing rate. And you see that rate directly from the neural activity are also positively correlated in animals. And also starting point Z was also significant in some of the neurons. And again, the neurons that encode this starting position was all part of the neurons encoding that slope. So there's also overlapping that as well. This is final data slide. We can also look at the high, population level trajectory. So you can think about linear speed defined trial start to trial end. You have this trajectory, and you can now break it down by low, medium, high, just to see if the evidence is high, are you going more straight path? Evidence is low, are you going to wind around a lot? In fact, that's what we find when we look at the path variability. So when path variability is high, the noise level was higher. That's why you see negative correlation. More interestingly, those correlation was only there when monkeys are actually successful pulling. So if there's a no trajectory, wiggliness relationship at all, then that was a failed cooperation. So this trajectory past variability related social evidence seems to be only relevant actually monkeys are doing the successful cooperation, which was interesting. And this is just another way to show you that uh, the variability was high because uncertainty is high, but then it goes down to shuffle level after trials, beginning of the trial. So just to conclude this section, cooperative strategies are highly flexible and tuned to social partner. DMPFC seems to implement this kind of gaze-dependent social evidence accumulation by transforming this active sensing, right, into your own decision variable to cooperate. I'm going to skip this one because out of time. I just want to thank everyone who did the work. The bolded individuals are parts of the study that I told you about. And I also want to highlight two co-PIs on the marmoset work we do in the lab. Thank you.